Good afternoon, everyone. I'm Leonardo Morelli. I'm currently a computational biologist for the Center for Omic Sciences at the San Raffaele Hospital in Milan. Here you can find my email. And today I'm going to introduce you to my master thesis project focused on the application of the nested stochastic block model to the analysis of single-cell data. Transcriptome analysis by single-cell RNA sequencing is a technology growing in popularity and application. In this scenario, identification of cells sharing similar properties is of paramount importance. A large number of approaches have been described, although the most prominent methods are based on modularity optimization. However, they share some important limitations. First, there is the lack of a method for evaluating clustering performance. The same algorithm with the same parameters usually produce slightly different partitions, but there are measures available to select the better one. Moreover, it is necessary to select an arbitrary threshold accounting for the size or the number of clusters. Finally, modularity optimization methods may identify community in random networks. In order to overcome such limitations, we have focused on the nested stochastic block model formulation implemented in the GraphTool Python library. This model is based on the assumption that a partition can be evaluated through the measure of the description length. This is an entropic measure which evaluates the amount of information necessary to describe data. Therefore, we perform cluster analysis through the minimization of the description length. In lay terms, we select the simplest model among models with the highest explanatory power. We developed schist, a Python library fully compatible with Scampi, to facilitate the adoption of the stochastic block model in single-cell analysis. Schist allows a statistical identification of cell groups and measures the confidence of group assignments. The nested stochastic block model relies on a robust statistical modeling. Therefore, the number of groups identified mirrors the amount of information comprised in the data. As a consequence, the absence of information can be properly handled. We collected data for 3000 PBMC available as processed data in Scampi. After standard processing, cell grouping is consistent in both approaches. When we shuffle edges of the pre-built nearest neighbor graph, in order to keep general graph properties unchanged, we found that the default strategies based on modularity optimization identifies 24 cell groups, while nested stochastic block model does not identify any cell group. The resolution parameter accounts for size of clusters identified by Leiden algorithm. The improper use of resolution parameter may lead to the identification of groups composed by two distinct cell types. To show this, we took advantage of public spatial RNA dataset of coronal section of murine brain tissues as provided by the recently introduced package SquidPy. At the full resolution, Leiden clustering resolved the tissue structure well, as does the first level of schist hierarchy. However, when resolution is decreased to 0.5, the dentate gyrus is incorrectly merged to the hippocampus, while schist correctly identifies pyramidal layer. The confidence of group assignments is estimated using the measure of cell affinity, based on the variation in description length produced by simulating changes in group assignments. We sought to exploit this property to address the task of annotating cells according to a reference sample. To this end, we analyzed datasets from the Humans Atlas project, which include mixtures of human PBMC and HEC293 T cells profiled with various technologies. We choose cells profiled with 10xV3 platform as reference dataset and perform annotation on cells profiled with MARS-seq. After having processed data according to the parameters given in the original project, we integrated MARS-seq dataset with 10xV3 into a unified representation using Harmony, and computed the k-nearest neighbor graph. We retained cell type annotation for 10x cells, while we assigned an unknown label to all cells derived from MARS-seq. Then, we calculated the cell affinity matrix. We computed the difference in entropy that can be observed by assigning each cell to each annotation cluster, this being either the original cell types or unknown. Once the matrix has been computed, each cell from the query data is assigned to the group with the highest likelihood. On the right, we compare the accuracy of the outcome to KNN classification given by the closest entry in the KNN graph and to ingest, a tool included in Scampi based on KNN classification and UMAP embedding. In conclusion, thanks to the entropic measure of the description length, schist enables the model selection. Second, one of the most important advantages of schist is the hierarchical definition of cell groups, which eliminates the choice of an arbitrary threshold on clustering resolution. Moreover, with schist, cell groups are accurately identified. Finally, confidence of group assignment can be calculated with cell affinity measure, which enables the label transfer. On the other hand, schist showed an increased run times compared to Leiden algorithm.